Target Malaria is an international, not-for-profit research consortium working to develop and share innovative and sustainable genetic technologies to modify mosquitoes and reduce malaria transmission. The project brings together several research institutions across Africa, North America and Europe. Today we will be visiting Target Malaria's insectary in London. Target Malaria's work here is led by Imperial College London, and the insectary is on the campus. Target Malaria started here as a university-based research programme and has grown to include scientists, stakeholder engagement teams, communication teams, risk assessment specialists and regulatory experts.
I'm Aleko Simoni, part of the Crisanti Lab, member of Target Malaria at Imperial College London. Our work here plays a key role in the project. This is where we design and generate genetically modified mosquitoes in order to generate an innovative technology for malaria control. Today, I'm taking you to visit our lab at Insectary.
The work at Imperial College is focused on designing the genetic modifications that are then introduced into a lab strain of Anopheles gambiae mosquitoes, which are the mosquitoes responsible for most malaria transmission in Africa. This genetic construct is designed and assembled in the lab, and then injected into the mosquito eggs in the insectary. The work done here is mainly assessing if the modifications introduced into the mosquito have the desired effect. For instance, reducing the fertility of the female mosquitoes.
This is the lab where we design and assemble the molecular components used to modify the genome of a mosquito. For instance, one of the molecular components we currently use to generate a gene-derived mosquito is CRISPR. CRISPR is a type of molecular scissor, or a DNA cutting enzyme. We use CRISPR to cut DNA at a very specific site in the genome.
Safety is paramount to Target Malaria. The insectary meets the international guidelines and national regulations for the containment of insects, called Arthropod Containment Level 2. Level 2 containment means that there are specific features in the lab built to ensure that insects inside the lab, in this case mosquitoes, cannot escape the insectary, and that external insects or animals cannot come in. Only a few team members have access to the insectary. Visitors must be accompanied by an authorised member of the team. Since we do not work with malaria-infected mosquitoes in our lab, visitors must confirm if they've been to a malaria-endemic country in the past 6 weeks. There are several security features around the insectary. These include badge access, a double door system, and an airflow machine. Entering the insectary is a 2-step process, and the first door can only be opened while the other is closed. This ensures that no mosquito can fly through the doors when they open. The insectary is kept at 28 degrees Celsius and high humidity to mimic the mosquito's natural habitat. A computer records temperature and humidity. An autoclave facility, which is a pressure chamber located within the campus, decontaminates and kills any waste. Once autoclaved, all waste is then incinerated.
Mosquitoes go through different stages during the development. First, the eggs are laid in water. You can see in this cup we have freshly laid eggs. Every little spot in this filter paper is an egg. The eggs then develop into larvae. You see in this tray there are the larvae moving around. Mosquitoes go through different 4 stages of larvae. And after the larval stage, they develop into pupae. You can see in this cup there are the pupae. The pupae is where the developmental stage of mosquito will develop into the adult form, which you can see there here in this cage.
Only female mosquitoes at their adult stage can transmit malaria because female mosquitoes bite humans to feed on blood to produce protein for their eggs. Female and male mosquitoes have different mouth parts. The female mouth parts are physically able to pierce skin. Males are not physically able to bite humans. We do not work with malaria parasites or infected females. To genetically modify a mosquito, we inject a DNA solution into the part of the mosquito embryo that leads to the development of mosquito germ cells. The germ cells are the cells that will develop into eggs and sperm in the reproductive organs of the adult mosquito. When introduced into the mosquito, this DNA solution produces enzymes that cut specific sequences of DNA and modify the genome. This is an extremely delicate and precise process and must be done quickly before the mosquito eggs dry out. When injecting, we must make sure we inject just the right amount of DNA. Too much DNA kills the embryo, and too little does not modify it. Together with the desired genetic modification, we also inject a gene that works as a marker, indicating whether the correct modification has taken place. For example, this marker could be a red fluorescent protein, and the eyes of the mosquito would glow red when looked at under a laser microscope.
Usually, a modification is inherited to only half of the progeny, and you can see in this case there is a green marker, a green fluorescent protein, and you can see that only half of the mosquito larvae are modified and the other half are not modified. This is because a gene is usually inherited only half of the times.
The injected enzymes are the core components of a gene drive. They work by identifying and cutting precise sequences in the genome, for instance in essential genes such as genes required for fertility. A gene drive is a genetic modification that is inherited among a disproportionately high fraction of offspring. Usually a gene is inherited in 50% of the progeny. A gene drive, instead, is inherited in up to 99% of the progeny. Therefore, over time, the gene drive mosquitoes can spread through the whole target population.
One of the gene drives that we are developing is targeting the fertility of female mosquitoes. That means that when those mosquitoes will be released, they can reduce the population size of mosquitoes in order to control malaria. We are setting those mosquitoes through a set of tests, and the next step of the technology will be to move those gene drive mosquitoes in Italy, where they can be tested in large cage facilities.
Once the eggs have been injected to introduce the modification, they are then reared to the adult stage. It is at larval stages that mosquitoes are screened for the genetic modification to check if the injection was successful. Larvae are screened using a special laser light. Mosquitoes with the genetic modification have eyes that glow. Once the larvae are sorted, they are separated and continue their growth cycle separately.
We can distinguish the males and females mosquito at the pupal stage. This is when we collect them and separate them under the microscope, and we put males and females in separate cages. This is important because we can in this way control mating.
Modified mosquitoes are then mated with wild mosquitoes to create and maintain a strain of mosquitoes carrying the genetic modification. These modified mosquitoes are then put through a series of tests. For example, we check how the modification affects the fertility of the mosquito, and we look at the transmission of the modification to the progeny across several generations. When the gene drive is successful, the genetic modification is inherited by more than 90% of the progeny. We conduct these processes again and again, generation after generation. Additional tests such as polymerase chain reaction, PCR test, or DNA sequencing are done to analyse the DNA of the mosquitoes.
A PCR, or polymerase chain reaction, is a technique that is used to amplify specific regions of DNA. This is important to check the correct integration of the modification we introduce in the genome, or it can also be used to study the expression of genes.
When mosquitoes die or are terminated, they are put in an autoclave and then burnt in an incinerator. We hope this tour has answered some of the questions you may have around our work. At Target Malaria, we are committed to being open and accountable as we work to develop an innovative tool for malaria control.